So let's look at this, the module 3 of 2022's biology paper, and look at reproductive biology. A student sets up an experiment to determine the ideal glucose concentration for germination by measuring the percentage germination of pollen grains in impatient, right? Right? So figure 4 shows an illustration of the experimental setup. Each petri dish contains different numbers of pollen grains with varying concentrations of glucose. So it's similar to that what occurred in 2017, right? It's generally okay. All right, it's really that. So state the suitable hypothesis for the experiment. What is the suitable hypothesis?
Sir, would we talk about like the higher the concentration of glucose, the higher the germination?
Okay, the percentage germination increases with percentage glucose. Not percentage glucose. With glucose concentration. All right. So we have that there. So what we're supposed to do is just interpret the thing and write down a nice suitable hypothesis. What is one possible error for this experiment? What is one possible error that you could possibly see? They could be varying pollen grains from different species, yes? What's a possible source of error?
Sir, could we also have like contamination?
In petri dishes, so if we have contamination, all right. So we have some amount of contamination that could affect the results. If the pollen grains are from, even if it's the same species, now if they are from different plants, that could cause an error. And each petri dish contains different number of pollen grains, right? So notice that, so each petri dish contains different number of pollen grains. So I think that is the best question, best answer. Let me put an asterisk. Each contains a different number. Where's the number side number of pollen grains, right? So wouldn't be able to to see the clear distinction. I think this is the best answer here. That's why I put an asterisk there. Because it's in the question itself, different number of pollen grains. State one change in the experiment that could have overcome the error stated. What is one change? So I think the best error would have been the that contains a different number of pollen grains. Just stick to that. Right? What's one thing that can fix that error now?
Adding an equal amount of pollen grains in each petri dish.
Okay. Pollen grains in each petri dish. Oh my gosh, how do I spell each? Okay, there it is. Alright, each petri dish. Alright, lovely. And name one responding variable. What are the differences between responding variables and independent variables?
Sir, could you repeat the question?
What are the differences between independent and dependent variables? I rephrase the first question rather. Go ahead.
Dependent variable is basically what you're changing in the experiment, and the dependent variable is what you're measuring. You just change of the
Huh?
independent variable.
That sounds weird. Mm-mm-mm. Mm-mm-mm. Hmm. The dependent variable is the responding variable, right? The dependent variable, right, is the outcome or the result that is being measured, while the independent variable, right, what would be the independent variable?
Oh, in this case,
Yeah, well, not in this case, right? But what would be the independent variable generally? What is an independent variable? In case it comes, what's an independent
what
variable?
is
Go ahead.
changing in the experiment?
It's a factor or condition that we control, right? That is manipulated in order to change or to allow for another variable to respond to, right? So our independent variable in this case would have been what? Our independent variable would have been our manipulation of the degree of glucose concentration, yes? That's our independent variable. That is what we manipulated. That is what the experimenter manipulated, right? So what is responding to that change in glucose? What is our dependent variable?
Sir, percentage of germination?
The
Percentage.
glucose concentration.
Wait, hold on. Is the glucose concentration changing in the experiment? No, no, no. No, no, no. That would not be accepted. Right? So the person said glucose concentration, right? In the experiment itself, right? Would glucose be the thing that is changing? Isn't glucose the thing that the experimenter manipulated? So if glucose is the thing that the experimenter manipulated, they put 0% glucose in this petri dish, then 0.002%, then 0.2%, was 0.02%, then 0.2%, right? So what we're doing is changing. That is what we manipulate. That is our independent variable, right? What is the dependent variable? No, that is what would be the response of our experiment, right? So our independent variable in this case would be the glucose percentage glucose. That would be what we manipulated. How we put different amount of glucose in a different petri dish. That is what the experimenter manipulated. While what would be the responding variable is the percentage germination that we see responding from the difference in glucose concentration. Right? So hopefully that makes a little bit more sense. It makes it a little bit clearer. The responding variable would generally be the percentage germination. Okay? So let's try to not mix it up. Whatever the experimenter manipulates, that is what we call the manipulated variable or the independent variable. And whatever happens, right? What is the response, the effects, right? The results, that would be the responding variable or the dependent variable in that case, in this case, right? State the function of the following structures as the answer. What is the structure of the epidermis? Structure, the function of the epidermis.
Sir, protection?
Somebody should make a meme about that. Every single time a biology student says epidermis, the first word is protection. Right? It's the first thing that comes. Protection. Alright, so yeah, protection. Right? So protection specifically. Like that's literally like the first word that comes up. As we say epi, we just write protection. Right? So well we shouldn't do that though. But epidermis there. What's the function of the tapetum, the tape tongue?
Sir, is it that it nourishes the pollen mother cell?
Mm. The
They
microsporangium.
provide nutrients, hormones, and other stuff for
Okay.
the mother cells.
All right. So we know that, right? So both of you guys are correct. Correct. Right? So it provides nutrients or nourishment or whoever you want to place it. Provides nutrients, right? For the microsporangium. Or what we call the mother cell. Right? But I like to use the biological terms that are given to us. Right? So it helps in that it helps protect it as well. Some protection as well. You could see protection. protection is there as well, right? It protects it from developing any type of stress, environmental stress, right? So it would have provide the support and nourishment in this micro environment, protecting it from any type of environmental stress, right? But that's just a secondary thing, right? What is the purpose of our microsporangium? What is the function of our microsporangium, or what we call our mother cells? Go ahead.
It undergo microsporogenesis to
Mm-hmm.
actually give rise it depends to four pollen.
It's going to develop into the pollen grain, which will have the generative cell nucleus and the
Mhm.
pollen tube nucleus. And
Lovely.
also the generative nucleus will undergo mitosis and it will give rise to the male gametes.
Microspora. So the microsporocyte specifically, right? So the pollen mother cells, right, from the microsporangium would actually now produce the microsporocyte, right, specifically, right? It would actually produce the pollen grains. So it produce the pollen grains.
Yep.
And now into my favorite parts. Let's go the steps, right? So sporogenesis, right? Our let's go with sporogenesis, the gametogenesis. What's the first thing that happens?
Some sort of mitosis.
Some sort of if you ever write some sort of mitosis on people them paper. If you ever some sort of
So
First, it undergoes meiosis.
so yes,
Pseudomitosis.
the mitosis would have occurred with the microsporangium to produce the mother cell, right? Right. So after that, now we already have it with the mother cell. So what happens through the mother cell now? The mitosis is done. Alright? So what now is meiosis?
um, it
First, it
undergoes
undergoes
meiosis.
meiosis, and
So meiosis one would give us what?
So
it
the
will
four
give us
haploid cells.
four haploid daughter cells.
Okay, wait, hold on. Meiosis one.
It's two haploid.
Right? So we're going to have two haploid. That's interesting. Right?
So it's not 4.
No, meiosis one and meiosis two, you know.
One. Yeah.
Meiosis one, the first division would actually give to two haploid, right? Secondary micro, right? Microsporocyte, right? Alright? And then meiosis 2. I remember there are two steps, right? Meiosis 2 now would give rise to the 4 haploid. Can I just write this? Because to write that again, the same thing really. You just give rise to 4 of them now, right? So we have that there, right? And then these now are called actually microspores. So I shouldn't really write that they're now called microspores. I mean, really and truly, they're the same thing. So microspores, right? So haploid cells called microspores. There are really 4 of them, right? And then with what happened now, they are combined within a tetrad, right?
Yes, sir.
Tetrad, yeah.
Combined in tetrad. And what would occur, right?
Eventually, they
Yeah.
separate.
Mhm.
Yeah, eventually,
So
and
tetrad.
then each cell
Mhm.
will undergo.
And
Then
each.
each cell will undergo
Mhm.
mitosis in the
So
cell nucleus, not the entire cell.
so we have what we call endomitosis, or mitosis within the cell. So nucleus splits, right? Yeah, splits to give rise to tube and degenerative nuclei. All right. So it's generally that structure, right? It's not too in depth, but it's generally that structure, right? Explain the significance of double fertilization in the embryo sac of a flower. The significance. What is the significance? One, we have the production of the zygote, right?
The zygote represents a new generation of
Mm-hmm.
species.
Well, not of species, but of like a new generation within the species. Don't say of species because they're gonna be like, what in the species?
Within the species. Yeah.
Yeah. So wording is important, right? So you can have a new zygote, yes. And what is the second
No,
thing?
sir, I meant within the species.
Within the species, yes. So what is the next one?
The triploid nucleus
Triploid nucleus, right? So we're gonna have the
is
The end.
in the endosperm, which can provide like nutrients.
mm-hmm. So we're going to have the production of the endosperm. Production of the
Sir,
yeah.
I was asking, the endosperm, does it has the ability to develop into the cotyledon,
Mm,
or is it just the same?
it remains more or does it develop into the cotyledon? Could somebody answer that?
No, it wouldn't.
Okay, so somebody said no.
Yeah.
Because remember, the endosperm basically it provides nourishment to the zygote as it develops. So once the zygote develops, then the endosperm will be depleted, basically.
Remember, it's the endosperm right, is the nutrient supply, right? So the zygote will develop into the embryo, and the embryonic hemispheres would now separate at a hypocotyl to produce cotyledons, right? So it's not the endosperm won't become a cotyledon, all right? So
Okay.
So in this question, I think the 3 marks come from you describing what happens in double fertilization, and then speak about how it produces zygote and how it produces endosperm for nutrients of the zygote or for the growing embryo. That's just 3 marks, right? Here now we look at a flow chart relationship among the hormones gonadotrophin, releasing hormone, right? Then we look at luteinizing hormone and follicle stimulating hormone, testosterone, and inhibin. Right, so I did go through this. There's a YouTube video of an explanation of all of spermatogenesis. So you guys could look at that. So using the information given in the flowchart, outline how the body controls spermatogenesis in the human male. How much marks? 4 marks. And should I slap on a nice 8 marker onto this, Or 15. I'm joking, not 15. Alright, so let's look at this. Explain it. Somebody come and explain it. They all give you negative feedback. They gave you all the information you need to answer this question. They even gave you more. I wouldn't, if I was setting the exam, I wouldn't even give you a diagram. I would just ask the question and ask you to write the flow chart. But go ahead.
So the hypothalamus releases gonadotrophin hormone, which is sent to the pituitary gland, which stimulates the pituitary gland, and the pituitary gland will release FSH and LH. So the LH will go to the Leydig cells in the testes to produce testosterone, and the FSH would go to the Sertoli cells in the testes, and that would cause spermatogenesis, right?
So wouldn't the LH also go to the Sertoli cells?
Would it also? All right, if
The
you
testosterone will go to the Sertoli cells to stimulate
yeah,
spermatogenesis.
so it helps. It helps stimulate the Sertoli cells. Yes. So the person who just spoke actually went through all of the main process now, but I think we need to now make reference to negative feedback loops. Who wants to have a go at that? So if we increase the prevalence of testosterone to a extremely high level, we're gonna have a negative feedback loop in which testosterone, right, itself will try to inhibit, right, the function of the hypothalamus and the anterior pituitary gland, as well as we look at the inhibin, right, from Sertoli cells who try to now inhibit um the the actual production of FSH and LH. So as we know within biology, within negative feedback loops, right, we see hormonal control in this way, in which we have the general structure, right, of hormonal flow within a system. But if it reaches to extremely high levels, right, the hormones produced ultimately will now lower the production of FSH and LH, right, which will now limit spermatogenesis, right? So what we're generally looking at is the fact that the inhibin and the testosterone can actually go back into the loop as a negative feedback mechanism, right, in order to lower the production of gonadotropin releasing hormone, right, as well as FSH and LH. So it regulates the amount of testosterone or the rate of spermatogenesis, right? It's regulating itself because it will produce hormones which will actually negatively affect the entire process itself, right? Right? So what we're doing, we're producing the hormone, but when it reaches a certain level, it's going to be so great, right, that the negative feedback loop is going to now overcome the original process and actually stop the release of gonadotropin, right, as well as FSH and LH. So it's just maintaining. It's like homeostasis. I hope you guys can remember the concept of homeostasis from system biology, right? If the hormone gets too much, then it's going to be slowing down the entire process. So it's speaking about the control of spermatogenesis, right? How does the body keep it under control? All right, and if there's too little, then it's not going to be enough to stop the release of gonadotropin and FSH and LH, so it's going to continue the process. So hopefully you guys can look at the diagram and explain it, right? So all we need to do is just outline how the body controls using this, right? So an organizational technique that you could use, even though this sounds very common to this, right? But remember, comes to this is just communication, right? So how could you communicate this to the person now, right? Some persons may list, because I would list. I would say, okay, step one, explain what I think in step one, right? So if I do it stepwise, it would help me to clearly denote what I'm talking about. If I put it in one paragraph, I have the higher chance of messing up the question. So in this case, they're asking to outline, so you could use different techniques to outline to make sure that you clearly see what you're talking about and you clearly see what you already spoke about. Because the minute you start to repeat yourself is the minute marks start take off, right? So you try to make sure that you outline it properly. All right. I don't want to go too long, and I want to stop at around 9 or 10ish. Right. So I don't do too much. Go ahead.
Sir, excuse me.
Mhm.
You were saying something about organizational techniques. Could you expound a little bit on that, please?
All right. So the organizational technique specifically within biology exams. In biology, we know that specificity, right, is something that we want. We love perfect specificity, but it must be specific to the problem at hand. So we need to analyze the question and make sure that we're using own examples and criteria that fit the question, right? But in areas where we have a lot of information, we're trying to make sure that we organize the ideas in such a way where it makes it clear to the examiner what you're talking about and makes it clear to yourself, right? Because if it's clear to yourself, you're going to have less errors. So what I'm saying here for the organizational technique for outlining, right? I would probably use a step by step, or I would use bullet points, so I can clearly read back what I write and then move on. If I use a paragraph, I might get mixed up. So organizing your answers and your responses is key. So that's why when they give you like the 15 mark, well, not 15 mark questions anymore, those don't exist anymore. Probably like a 12 mark or 8 mark question describing a process. It's good to write the name of the process and then you use your colon and you describe the process. Try to list them out neatly so you have your ideas on the paper properly, right? Because
Okay, sir. Thanks.
some people tend to write in one big paragraph and then realize in sentence four they might talk about the same thing in sentence one and mix up. Right? Big ball of confusion. We're trying to make sure that we avoid the confusion in paper. Alright, so D, there is increasing evidence that mother's behavior during pregnancy affects the development of the fetus and can cause long-term health consequences for the child. Figure 6 shows the predicted effects of maternal smoking on birth weight based on the population study in British Columbia between 2001 and 2006. Alright, we're looking at that data there. Using the information in Figure 6, discuss the effects of maternal smoking on fetal development. You must refer to Figure 6 in your discussion. You must. So what's happening here with the birth rate, the birth weight rather, and cigarettes per day? Right, so we're seeing the more cigarettes the mother have per day, right, the lower the maternal birth weight. So what is that? What is the effect on not maternal birth, well, the fetal birth weight, right? So what is the effect we're seeing here? Somebody tell me.
Sir, could you repeat the question?
All right. The question is asking, right, to discuss the effects of maternal smoking on fetal development, right? So in the graph, now we're looking at birth weight, right, and cigarettes per day. So now describe the graph, explain the graph to me, right? We're looking at the fact that the increased amount of cigarettes per day decreases the birth weight. So now speak to that, discuss that, and discuss how the smoking affects the fetal development. Go ahead.
Okay, sir. Would you say like the less cigarettes you smoke per day, the higher the birth weight, but as you increase the amount of cigarettes you smoke per day, it lowers the birth weight?
How? How so? I love to ask those questions, you know. How so? We see it in the graph, you know, but how so? What is happening?
Sir, carbon monoxide, nicotine in the cigarettes.
Not necessarily nicotine, but plaque, right? Tar rather, and your carbon monoxide is important. So what is happening to the fetus as you increase smoking?
It's getting less oxygen.
It's getting next oxygen. Alright, so when you have hypoxia, low levels of oxygen, we have fetal hypoxia. Lovely. And that is due to probably the tar in the lungs of the mother, right? Reducing oxygen circulation. And what would the smoking also do to the umbilical blood vessels? What would they do to them?
Make them smaller.
So it would constrict the blood vessels. Lovely. So this is how we should approach questions now. We're talking about the reduced placental blood flow, and we're talking about fetal hypoxia, right? And how those can actually affect the developmental rate of the children, of the fetus, right? Can affect the developmental rate of the fetus. So when the fetus is now born, they're underdeveloped, as seen by the lower birth weight, because of the lack of oxygen, the fetal hypoxia, as well as the reduced blood flow, reducing the nutrients and everything that the fetus may need. Right? We could look at even the use of tar, nicotine, and various carcinogens from the cigarette that would lead to toxic chemical exposure to the fetus, and the impaired nutrient absorption. So there's not going to be proper nutrient absorption, and therefore there's not going to be leading to proper development. Therefore, your child will be born underweight. That is what we must talk about. 4 marks. Right? A lot of information for 4 marks, yes? Talk about it, man. We have the information, use it. Alright? A pregnant woman visits an antenatal clinic and is advised by the physician that as a result of her lifestyle, the baby has a high risk of having a low birth weight. The physician strongly recommends that she makes lifestyle changes that can prevent this outcome. Discuss two maternal behaviors other than smoking that may contribute to low birth weight. You must include one lifestyle change that the physician may recommend for each maternal birth behavior discussed. What are other behaviors that can contribute to lower birth weight?
Drugs.
Okay, be more specific. Specificity is your friend. Let's go. Drugs, what drugs? Mm. Give me one moment. All right. Mm-hmm.
Illegal drugs such as methamphetamines and amphetamines, which increases the risk of low birth weight and premature birth.
Okay. Methamphetamines and stuff like that. Okay.
Sir, what about diet? Like eating food that contains folic acid.
Well, not eating. Okay, so in this case, we look at the behaviors that are not right. So, if they have poor nutrition, inadequate intake of certain nutrients, right, then that will lead to low fetal development and low birth weight. Lovely. And the other person mentioned the use of different types of drugs, right? And stuff like that, like methamphetamine, etc. Right? Those different types of drugs may inhibit fetal development, right? What else could happen, right? You could have maternal conditions. So if they have pre-existing conditions like diabetes and hypertension, those type of things, that will lead to a low birth weight in the fetus as well, right? Could speak about that as well. So not only the use of illicit drugs and alcohol can lead to different types of issues within the mother of a low birth weight, right? But the nutrition, the lack of nutrition or undernutrition, right, as well as stress. Stress can contribute to low birth weight as well. All right. So what are some lifestyle changes? So for the poor nutrition, we'll increase the amount of useful vitamins by using green leafy vegetables will contain mostly our folic acid and our vitamin E, right? And these different things, right? So we're increasing the nutrients such as proteins, vitamins, and minerals, right? Less carbohydrates. So if you eat a lot of carbohydrates, that could lead to hypertension. Note that. The use of illicit drugs and alcohol, all we have to do now, what would be our lifestyle change, right? Reduce the consumption of alcohol and the use of illicit drugs. That makes sense, right? That's very straightforward, yes? Question is very straightforward, honestly. Wait, how many marks is this? I don't see the marks for this, but it's a lot of writing. Alright. But that is it for Module 3 of the 2022 paper.